We have Dana Vanderwall from Bristol-Myers Squibb. He's going to talk about the usage of instant JChem for patent analytics.
Okay, thanks. Thanks for having me, guys. Thanks for the hospitality, the entertainment, or in some cases, the torture. So actually, you've heard a couple of stories about patent analytics, document markup, annotation of documents. And it's something we got involved in at BMS a few years ago in the context of what was at that point a collaboration with IBM Research being led by Steve Boyer, and some of the other folks here have been involved in that. But one of the things we haven't talked about yet today is how do you really get your head around that, right? So it was a watershed moment to get access to full text patent documents and pull all the chemistry out. But then it's kind of like opening that closet that you've just been kind of shoving things in over the years, and you open it up and it all falls on top of you. So that's sort of the tack that I want to take today. It's less about technology and it's more about an approach we've taken trying to utilize this data or help end users utilize it, really, your average guys in the lab. So just as a brief introduction, right, you've heard a little bit about this. I stole this slide from Steve. Some of you guys have probably seen it before. But basically, the overall idea here is that there's a curation factory or a computational approach to annotate patent documents and things from Medline as well. I'll focus on patents today. But there's been on the order of 15 million patents loaded into this machine, right? And they're parsed. All the structures are captured. There's 3 different ways of capturing the structure via the name, and one of those is, of course, the ChemAxon named structure. The mole files, or the chemical work units that are submitted, at least for now, with U.S. patents, and then also using the chemical structure equivalent of the OCR, the OSRA, getting the images. But the end result is this database with XML versions of the patents, marked up for where the structures are, where the gene IDs are, things like that, and indexed, right? So indexed by a text indexing engine that sort of breaks out all the individual pieces of the structure of the patent, all the different sections. And for most of the collaboration, most of the access was by way of a web application that was sort of developed along with the rest of the infrastructure here. But there's also the possibility to use a copy of this data inside our firewall. And there's a couple reasons to do that. One is that, we have maybe yet to develop a comfort level of doing IP-sensitive searches over the web, even on HTTPS, right? But the other is with those structures in-house, there are other ways that we can leverage them other than just through that particular application. So the kinds of questions that we wanted to help the scientists address are, some of the fairly obvious ones, but basically being able to search the patent corpus by chemical structure and different versions of that, right? We want some flexibility around that. To answer questions about whether there's prior art, whether the things you're thinking about making or that you've made are patentable, to get a sense of what the competitor's activity is around either a chemical series or ultimately around a therapeutic area, target area. And one way we've been using this sort of thing, and it's been inferred a little bit, is sort of as a surrogate profiling, if you will. So if you have molecules for which you have some data, maybe not as much as you'd like, to understand what their target profile might be, because you haven't screened them on the genome yet, another possible scenario is searching the patent database, find out if you've got reasonably similar molecules in the patent corpus that have particular therapeutic areas, indications associated with them, particular target activities. And so that gives us sort of a surrogate measure of what the activity of that compound might want to be. It's the poor man's version of the very sophisticated Bayesian approach that John was telling us about earlier to some degree. And then you want to be able to show and browse the chemicals in a patent. And for this, it might be nice to find some sort of a paradigm for visualizing this stuff on a screen that is more user-friendly than just flipping through pages and marking them up, right? There's some really interesting questions once you can get this data out in different formats and thinking about how you enable tracking more effectively a chemical series throughout a series of patents over a function of time associated with a particular company or with a therapeutic area. And then finally, we want to be able to do some of these bigger data mining exercises where you just suck out all the structures, you look at the indications, you look at the titles, and you look for patterns, for example, in the chemical space associated with a particular therapeutic area. Or you compare your compounds in a therapeutic area with the stuff that's in the patent literature. And so you can ask these kinds of questions with chemistry overlap. But for that, you really just need to be able to get your arms around all these structures at once. So those are the kinds of reasons we wanted to bring this in-house. For the primary use of the end users, we're leveraging instant JChem. And this sort of falls right in with our overall plan, which is to move everything we do in drug discovery analytics, all the data access, all the analysis from our existing application and infrastructure into instant JChem. Of course, that could be a whole other talk. We're not going to talk much about that today, but we'll spend the next 3 years over this transition. But in the meantime, it's actually a simpler problem to plop instant JChem on top of this IBM patent database relative to sort of going around talking to all these program teams. So this gives us an opportunity to use this in-house instance of data. And one of the advantages is because this is going to become the BMS standard for discovery analytics, now the chemists don't have to go to a separate application to go search the patent literature. It's just another project over on the left-hand side. So it brings more things under one roof. And they can do these searches, the chemical structure searches, search by a patent, or by the text searches in the same application. The other is that while it's still evolving, the instant JChem platforms is still a pretty good visualization paradigm for chemistry and for scientists and drug discovery in general. Just the kinds of layouts and the interactions between the tables and the grid views and things like that. So we wanted to be able to leverage that to help people sort of digest this data. And finally, we want to move our current practices in patent analytics and supporting drug discovery teams from something that's very document-based, which includes searches in the old tools that everybody's been using, paying by the compound to download compounds, pasting them all into a Word doc and sending them off. And now they're sort of static and you have to redraw molecules. To something like what we can do in Instant JChem, where somebody in the patent analytics group does some work, does some searches, captures the result of that as a URL in IJC and sends that off to the chemist. And they can sort of send these things back and forth to one another. So to get this interacting and collaborating a little more smoothly than these static documents. So this is just a couple of screenshots of what it looks like. And when we first started talking to people in-house about, yeah, we're going to use Instant JChem for patents, and they said, oh, you mean those big text documents? Well, how's that going to work? That's not going to work out very well. But actually, it's obviously it's not bad because you've got the sections of the patents. You're not going to stream all of the text in one box, right? You've got, depending on the kinds of questions that you're asking and what those threads are, what those workflows are, it's relatively easy to say, okay, in this particular case, they're going to want to see the list of patents first and the grid of molecules, the abstract, and the claims over here. They don't need the details in this particular use case. So you can really tailor the presentation in that sort of a way. It's a vast improvement over flicking through hundreds of pages of paper or just sort of streaming down long lists. So the variety of grids and views is we've made a lot of use of that. Gives us a lot of flexibility in the presentation. And then the scripting hooks that Aaron touched on are something that we're really just starting to take advantage to leverage something else that comes along in this IBM patent data. And Steve's mentioned it earlier, and that's the solar indexing technology. I'm not going to read through all this. We've talked about it a little bit, but here's some information for reference. But essentially, once you've indexed that corpus with the solar technology, we've got very fast search access to all these fields that are in the patent document, and very fast text searching within those fields. And that's one of the things in the original web app that's sort of built right in that worked very well. And that's not something we could access directly from Instant JChem. But that's where the scripting comes in handy. So Dong Lee, who I should mention, has done most of this work, now has put up this script interface. And for any of you who've used PatBase, this might look kind of familiar because it really actually looks a lot like PatBase. But now the users, they click a little button, this interface pops up, and there's drop downs for most of these fields, the ones that are most commonly used, the ability to string those together in complex Boolean logic, and some of the most popular combinations of the fields are already there. And then that takes the text that the humans type in and tortures it into the Solr syntax that you need to actually search the Solr database. And then you can pick the collections you want to search. But you click that, and off it goes, and really very quickly comes back with this version 1 of the report output. So this is just another window that gets popped up in Java Swing. The basic table. Here's the title, here's the patent number, and the assignee. So after a search, this gives the scientists or the patent analysts, an easy way just to sort of scan through and say, well, am I getting anything close to what I thought I would with this text search? But more than that, now that's been wired in to populate in the list manager that list of patent numbers that were found from that search. That goes back in to the IJC application from the script, so that now you just click on that list and boom, you're back into the typical forms environment where you can look at the summary level, look across those fields, or start drilling down into detail into each of the patents, look at more of the detailed information, look at all the chemical structures. So in this sense, the Groovy scripting is proved extremely powerful. And it's not in place yet, but the next step now is actually to merge the chemical structure search with the Solr text searching. And finally, I was just going to touch on a couple of little ways, other ways that we've leveraged the patent content using some of the ChemAxon tools besides Instant JChem. And sometimes the simple little things you do for somebody can really make a huge difference. So one of the things Don put together was just this little macro, right? And what it does is calls a web service, gives it the patent number, brings back the chemical structures, pops a new worksheet in the Excel workbook I don't remember if you use the smiles or the moles and then convert them, but basically puts all the structures on the worksheet using JChem for Excel, but then also leaves this, an HTML link to just a pop-up HTML grid, which of course is also used in JChem on the back end for that. So those HTML links are portable. Then you can put them in any document, and they point to that same service to pull up the structures. Another one that I don't have a picture of because it's just command line, and command line isn't much eye candy, but there's a command line version that mostly our CAD friends use. So for running some of the bigger queries, searches, batch things against the same database to pull stuff out. So this was all sort of an experiment. Let's try it in Instant JChem. Let's just continue to evolve it. We had a couple of milestones that we wanted to enable, including the text searching, including finding out whether we really still needed a full patent text browse that we had in the web app. Actually, I'm sorry, I remember one thing I didn't show here. There's also a URL now that links back out to the PAT-based full text document. So even though it's not interactive, if somebody really needs a full document, they can pull it down. But really, in a short time, this has significantly changed the way our patent analysts are working. This has become the tool of choice now, particularly for structure-based searches, and increasingly now as we add the text in for text-based searches. We also utilize this content with the Lingomatics I2E platform. So there's another little tool associated with this that, given the patent number, sort of pulls out the relative patents, conditions them and pads them and fixes them up a little bit for I2E and indexes them there. So whereas the old model of patent analytics really relied on an expert interface used by patent analysts, and there's a small team, and everybody in drug discovery who needed help had to go to them. It relied on high quality but really low volume human curation data, and really data mining the entire chemical space that we could suck from the patent literature was not available. That wasn't an option. And everything was on static documents, things that were stale given the way some, an active chemistry space or patent space changes, stale within a couple of months. And the access to the chemical structures, without going into detail, frankly, was just not cost effective. So this enables now more user-friendly access. Any of the chemists and scientists can go access this and do a reasonably good job. Still consult with the patent analysts when they need to, but now be able to do this in a much more collaborative way. And it changes, the rate at which people can make decisions or start to get a handle on a patent space when they start working in that area. And it's made some of these data mining questions feasible. And the platform itself has turned out to be very nice to do some, it's not really agile development because we're not doing that much development, but it's given us some agility to continue to evolve these forms and scripts and things as we go over the last 2 or 3 quarters. So it's been great. So finally, just some acknowledgements here. As I mentioned, Dong Li is the one who did most of the work. When we give talks, we always use the word we, but in this case, I didn't really do anything. It was all Dong. So the collective we was Dong in this case. And Steve Boyer, of course, at IBM is always fun to talk to, great to bounce ideas off. It's been helpful. And these other folks at IBM have been terrific, helping us understand how to better leverage our in-house version of this data and how really to optimize our performance against it. I should add that we have no fear at searching, that database of 9 million unique molecules. It's just, the performance of the cartridge is such that works well. The first time you do a benzene, if you haven't cached the search for a while, that runs for a little while, but the subsequent search of the benzene ring's fine after that. And then everybody at ChemAxon has been terrifically helpful. Tim, Aaron, Peter, and Slard on helping us get going on this. Thank you.
Nice talk. Have you thought of donating back the instant JChem schema to Steve Boyer, so maybe other IBM data customers can benefit?
I see no reason not to.
Yeah, that's the IBM on-premise provides the data. In the past we live on the SignFinder, and we know once the data on the other side of firewall is published. So, and I think we should donate some of the contribute.
Yeah, your views, for example.
Yeah, I think that
I don't know. You want the views, John? Huh? Yeah. All right. Sure. Well, that in the script, actually, for that matter, we can put that in the example scripts.